So we're going to start, and it's my pleasure to ask my co-chairman, John Allen, to have a first presentation about the effect of kinase inhibitors on motor function in Lewy body disease.
Thank you, Jacques. My name is John Allen. I'm CEO at EIP Pharma. I would like to first actually thank the scientific community, scientific committee to give me to provide us the opportunity to present to you the final results of the AscenD-LB clinical trial in dementia with Lewy bodies. These are our disclosures. I will also disclose that we are based in Boston, 2 blocks away from here. So as we are still very early in the meeting, I want to welcome everyone to our hometown. So, and then otherwise, I would like to upfront acknowledge all the people who were involved in this clinical trial, in particular the participants and caregivers, and then all the participating clinical principal investigators and their staff. As background, Neflamapimod is an oral small molecule drug that targets pathogenic mechanisms in cholinergic degeneration, and in clinical studies addresses both functional and neurodegenerative aspects of basal forebrain cholinergic degeneration, which is a primary pathology in dementia with Lewy bodies. At last year's CTAD, we presented the initial results of a placebo-controlled phase 2 trial, the AscenD-LB study, in mild to moderate dementia with Lewy bodies, in which Neflamapimod at a dose of 40 milligrams TID demonstrated a positive effect on the primary endpoint, a DLB-specific cognitive test battery, as well as on a secondary endpoint, the timed up and go test. For more background on the scientific rationale and why we're specifically targeting the basal forebrain in dementia with Lewy bodies, I would encourage you to go to the digital platform when you have time and view Symposia RS3, which is on basal forebrain cholinergic degeneration. But this is one slide from that symposia from Ralph Nixon, which shows that Neflamapimod reverses disease progression in the basal forebrain in transgenic mice that develop pathology in the basal forebrain. That preclinical study gave us the understanding that Neflamapimod could increase basal forebrain cholinergic function. And based on that, a group of experts designed a primary endpoint that would measure what at the time was considered to be the primary activity and function of the basal forebrain cholinergic system, cognition, in terms of attention, executive function, and visual learning. As presented at last year's meeting on this primary endpoint, there was a significant and clinically relevant effect in the 40 milligram 3 times a day dose group with an onset of at week 4 that was maintained out to the end of treatment. There was also a significant effect with a lower p-value when the components of the cognitive test battery that were specifically looking at attention were evaluated, as shown on the right-hand side of this figure. Subsequent to that presentation, there have been a series of publications, most of which came out in the first half of this year, that pointed to the basal forebrain cholinergic system also having a significant role, also in addition to the cognitive control, a substantial role in the control of gait and more generally motor function in DLB and Parkinson's disease. Given that understanding, we went back to the AscenD-LB clinical study to understand better the effects of beyond the cognitive effects, which was our initial focus, and specifically in terms of motor function. The AscenD-LB study was a 16-week placebo-controlled study in mild to moderate dementia with Lewy bodies by consensus criteria, including the requirement to have an abnormal dopamine uptake by DAT scan. Importantly, all patients in the study were on background cholinesterase inhibitor therapy. The study utilized a weight-based regimen, the intent of which was to achieve a uniform plasma drug concentration across all Neflamapimod recipients, allowing the group to be considered a single dose group and to be compared against placebo. However, while the 40 milligram TID dose group did achieve target plasma drug concentrations, the 40 milligram twice a day group did not. As a result, the initial analyses, the ones we presented last year, were focused on analyzing the 40 milligram TID group versus placebo. However, in the final analysis, because we were looking beyond where we saw the cognitive effect, analyses were focused on also analyzing the comparison of all Neflamapimod patients against placebo, and that which included the 40 milligram BID that was therapeutic in terms of plasma drug concentrations. And we'll also then show the 40 milligram TID, the high dose group, against placebo that achieved target concentrations versus placebo. These are the baseline disease characteristics which showed that Neflamapimod and placebo were well balanced, as well 40 milligram 3 times a day was balanced against placebo. The timed up and go test is a validated measure that measures functional mobility by monitoring the time required that a subject takes to rise from a chair, walk 3 meters, turn around 180 degrees, walk back to the chair, and sit back down while turning 180 degrees. As such, it is a test that comprehensively assesses gait, including initiation, quality, and speed, as well as balance. In AscenD-LB, the time required to complete the TUG test was balanced across the treatment groups with a mean of approximately 13 seconds, which goes along with mild but definite abnormality at baseline. And the use of carbidopa-levodopa was not a confounding factor. These are the results on the timed up and go study test. Again, last year we showed the data in terms of 40 milligram TID versus placebo, which is significant. The new analysis here is looking at all Neflamapimod patients versus all placebo patients, so the entire treated or the full efficacy analysis population. Despite including the 40 milligram BID twice-a-day participants, there is also a significant difference in this comparison favoring Neflamapimod when comparing Neflamapimod versus placebo. Because we did not see a significant difference in the all versus all comparison for the cognitive endpoint, the result on the timed up and go test from a statistical standpoint is more clear-cut. Given that result, particularly in the all versus all comparison, we went back and also did further analysis on the CDR sum of boxes, analyses that had not been conducted previously. Here again, in the all versus all comparison, there is a significant difference with a 65% reduction in the mean change of the worsening from baseline to week 16 in Neflamapimod compared to placebo. And then in the comparison of 40 milligram TID versus placebo, the p-value is 0.007. The particularly good statistics on the CDR sum of boxes, we believe may reflect that the test is able to capture the positive effects of treatment on both cognition and motor function. In the last part of the presentation, I will briefly cover some results from a poster presentation at this meeting, in which the hypothesis we wanted to test is to see if the beneficial effect of Neflamapimod in this AscenD-LB study was through improving basal forebrain cholinergic function. If it were that, it should be most effective in patients where the pathology is confined to the basal forebrain and more effective than in patients who have significant pathology in other regions of the brain. This is effectively doing what Ron Peterson in his talk at the opening session was talking about, looking at patients by whether they have mixed pathology or not, and whether in the patients where your mechanism of action aligns with the patient population you are studying, whether you do see a specific and a particularly strong clinical efficacy. And indeed, to test that sample, we do see that, and to test that hypothesis, baseline samples were assayed for plasma phosphatide 181, which in a publication earlier this year was shown to predict the presence of AD copathology and was particularly well correlated to temporal lobe tau pathology. In the AscenD-LB study at baseline, a little over 50% of patients did not have elevated plasma phosphatide, and thus would be predicted to not have AD copathology and therefore have disease confined to the basal forebrain. And the efficacy in this group is particularly good and appears better than those who have AD-like or temporal lobe tau pathology. I'm not going to go through all of this data. Again, I will encourage you to come by the poster that's LP14, but I will show you in an extension of the rest of the presentation the data on the timed up and go study test and as well on the CDR sum of boxes. On both slides, in the gray bars are the results from the MMRM analysis when the patients with low plasma phospho-tau and therefore have no co-pathology, the difference in that group relative to the placebo group, either for the Neflamapimod versus all Neflamapimod versus all placebo or 40 milligram TID versus placebo. Then on the timed up and go test, for either comparison, there is a substantial difference relative to placebo in these patients, a 2-second difference for Neflamapimod versus placebo with a p-value of 0.02, and a 3-second difference for the high dose of 40 milligram TID versus placebo with a p-value of less than 0.001, despite a relatively small sample set, which is a rather striking result. Similar results on the CDR sum of boxes, where the p-values are a little bit higher versus given the sample size, but again, a very obvious treatment effect in the patients with plasma phospho-tau 181 below the cutoff, and that is the patients without co-pathology, and a significant difference with a mean 0.6 point difference in the comparison of 40 milligram versus placebo, which is a large effect for CDR sum of boxes in a 16-week study. In summary, in the final results of the AscenD-LB study, the clinical activity of Neflamapimod appears to extend beyond the cognitive effects that we had presented at last year's CTAD meeting. Within the final results, Neflamapimod led to significant dose-dependent improvement versus placebo on cognition, on functional mobility or motor function, as well as on cognition and function, effectively dementia progression. These clinical effects, including particularly the better effects in patients without AD co-pathology, is consistent with substantial improvement in basal forebrain cholinergic function. To conclude, relative to placebo, Neflamapimod improved cognition in this study, motor function and cognition and function, or dementia progression, in mild to moderate dementia with Lewy bodies, consistent with a disease-modifying effect on basal forebrain cholinergic degeneration. The positive effects on functional mobility, the TUG test, provides further evidence of the role of the cholinergic system in the development of gait dysfunction in Lewy body disorders. And from a future clinical trial standpoint in DLB, in dementia with Lewy bodies, both the TUG test and the CDR sum of boxes appear to perform quite well to assess and evaluate and pick up drug treatment effects. Thank you very much for your attention.